Now that we have discussed a specification of the dorsal ventral axis, let's look at how the different regions across the anterior-posterior axis are specified. In other words, let's look at the factors that allow the formation of different structures from the head all the way down to the tail of the organism. And we will focus on the patterning of the nervous system across the anterior-posterior axis. Now, the specification of the anterior-posterior axis depends on a gradient of Wnt proteins, with the head having the lowest levels of Wnt, and we'll look more at that later. Now, in vertebrates, the forebrain, the hindbrain, and the spine regions of the neural tube have to be properly organized in an anterior-posterior direction. And the organizer tissue not only induces the neural tube, but it also serves to specify the different regions of the neural tube. So this happens in a region-specific way so that the more anterior portions or tissues that are derived from the organizer will induce the formation of more anterior neural structures, while the most posteriorly derived organizer tissues will induce the formation of more posterior neural structures. So transplanting 4 successive regions of the archenteron roof plate of late gastrula nude embryos, which are what's shown here, into the blastocele of other nude gastrula, it was shown that the most anterior portion of the archenteron roof, which contains the head mesoderm, which is the anterior portion of the organizer, induces the formation of balancers and oral structures, which are located at the most anterior part of the organism. The next most anterior section induced the formation of different types of head structures, including eyes, nose, and balancers. The third section, which includes the notochord, induced the hindbrain structures. And finally, the most posterior section induced the formation of the dorsal trunk and tail structures. Recall from the last lecture that the identity of the dorsal blastopore lip changes over time as the more anterior structures begin to involute during gastrulation. So if you were to transplant a dorsal blastopore lip from an early gastrula from one salamander to another, you would end up with a salamander that has a secondary head. Whereas if you were to transplant the dorsal blastopore lip from a later salamander to an early salamander gastrula, you would end up with a salamander that has a secondary tail. This is because in the early gastrula, the blastopore lip is composed of the anterior endodermal and mesodermal structures, which induce the anterior ectodermal structures, but later the blastopore lip is going to be composed of more posterior mesodermal structures, which will induce the formation of more posterior neural structures. Now, these results show that the first cells of the organizer to enter into the embryo induce the formation of brains and heads, while those cells that form the dorsal lip of a later stage in development are going to induce the cells above them to become spinal cords and tails. The question then became, what are the molecules that are secreted by the organizer in a regional fashion so that the first cells that involute through the blastopore lip or the endomesoderm induce the formation of head structures, whereas the next portion that involutes through the mesoderm, which would be the notochord, would produce tail and trunk structures. Now, the leading edge of the dorsal blastopore lip, or the first tissue to involute during gastrulation, is a tissue that becomes the pharyngeal endoderm and the precordial plate mesoderm, and these together are known as the head endomesoderm. So in addition to secreting BMP inhibitors, these tissues also secrete inhibitors of the Wnt signaling pathway, and these are the inhibitors that you see here. Now, blocking the Wnt pathway is necessary to allow the formation of the head. So in addition to having to block BMPs to allow the formation of neural structures, Wnt's have to be blocked to allow the formation of head structures. So in the regions where the head structures form, both BMPs and Wnt's are blocked. In the region that will become the spinal cord, BMP inhibitors are secreted by the organizer, but this region lacks Wnt inhibitors. And finally, where Wnt inhibitors and BMP inhibitors are absent, the ectoderm becomes specified as epidermis. Since inhibiting Wnt is important to allow the head to form, if you were to overexpress Wnt inhibitors, the embryo ends up with extra heads or ectopic heads. In this image, an embryo was injecting with an mRNA for the Wnt inhibitor Severus, and as you can see, the embryo developed an extra head as a result. Now, if you were to block Wnt inhibitors like Cervarus, then the head would not be able to form. There are a number of different Wnt inhibitors, which include Cervarus, Frisbee, Tiki, and these are synthesized predominantly in the endomesoderm cells beneath the prospective brain. In embryos that overexpress Frisbee, Wnt signaling is blocked throughout the embryo, and the embryo becomes all head and lacks all posterior and all ventral structures.